Okay, assalamu alaikum. Today we are going to study the structure of mitochondria and functions related to the aerobic respiration that is involved in order to generate ATP. So, in eukaryotes, what does the term eukaryotes mean? Eukaryotes are those organisms that have a proper true nucleus, or we can say they have a well-developed nuclear membrane. So, eukaryotes, in eukaryotes, it is a site of Krebs cycle. Krebs cycle is the third stage of aerobic respiration and electron transport chain. These two processes are a part of aerobic respiration that are used to generate ATP energy-rich molecule. So, mitochondrial structure, the mitochondria is a site of Krebs cycle and electron transport chain in eukaryotes. Now, coming to the shape of mitochondria, it is either rod shape or filamentous organelle. So, it looks like somewhat like this. It is a rod-shaped structure. It has an outer membrane, inner membrane, and then mitochondrial matrix. This is an outer membrane. Then it has a folded inner membrane, and then cytoplasm, which is called matrix. Now, what is the size? The size of you may have studied this in your AS bio, that the size of mitochondria is approximately 0.5 to 1 micrometer in diameter. Now, the number of mitochondria depends on the activity of cell. The much a cell works, it should have increased number of mitochondria, because as we know that being a biology student, we know that mitochondria is also said to be the powerhouse of the cell that generates energy for each and every cell. So, as much as a cell works, it needs more amount of energy. To have more amount of energy, increased amount of energy, it should have increased number of mitochondria. So, as much as a cell is highly active, working, it contains the more number of mitochondria in it. An example of it is so an example of it is highly active mammalian cells, which is liver cells. Liver cells are highly active. They work constantly to remove toxic substances from your body, as well as they perform the function of glucose storage and much more. Formation of vitamins, fat-soluble vitamins. So the liver cells continuously do work. So we can see that the liver cells contain around 1,000 to 2,000 mitochondria in each cell of liver. So each cell of liver or liver cell, which is also known as hepatocyte. So each hepatocyte, which is a liver cell, contains 1,000 to 2,000 mitochondria in it. That is, the 20% of the cell volume is occupied by mitochondria only, and then the rest containing other organelles. Okay. So mitochondria is a similar structure to an organelle which is found in plants, that is chloroplast. So like chloroplast, it has two envelopes of phospholipid membranes. The outer one is called outer membrane, which is quite straight, linear, and the inner membrane is much folded. The folds of inner membrane, these folds are called cristae. So the outer membrane is quite smooth, whereas the inner membrane is folded inwards, like this. This is the outer membrane, and then here we have the inner membrane, it folded inwards. These folds are termed as cristae. So what is the significance of these cristae? That they provide a large surface area, and you must know that this inner membrane, the folds of the inner membrane, they are a site of electron transport chain. The proteins in the electron transport chain, they are present on the inner membrane. So as much as the large area we can have more proteins accumulated in that, so that at the same time we can have more electron transport chains taking place and forming energy-rich molecule ATPs. And then the permeability of the outer membrane is that it is relatively permeable to small molecules. So outer membrane is more permeable to small molecules, whereas the inner membrane is less permeable to molecules, whether it is small or large. So inner membrane is studded. The special characteristic of the inner membrane is that it is studded with tiny spheres, which is about 9 nanometer in diameter. And these spheres are basically ATP synthase enzyme, which forms, whose function is to synthesize ATP. So in the on the inner membrane, we have tiny spheres, which is about 9 nanometer in diameter. And these spheres are ATP synthase enzyme, whose function is to perform the synthesis of ATPs. The inner membrane is a site of, as we discussed earlier, it is a site of electron transport chain, and it contains proteins that are required for electron transport chain. Now the space between the two, that is between the two membranes, has a lower pH than the matrix of mitochondria due to the release of protons in the intermembrane space by electron transport chain. So what happens is that the proteins embedded in this inner membrane, what they do is that they transfer protons from the inner membrane, inner space, into the matrix. So the matrix has a higher pH rather than the space between the two membranes. So this space between the two membranes, the concentration of proton is much less, as the proteins in the electron transport chain, they are transporting this protons which are present in between the two membranes into the matrix of the mitochondria. So the matrix of the mitochondria has a higher pH rather than the space between the two membranes. So talking about matrix is the site of link reaction. You must have studied that a link reaction takes place in the matrix of the cycle, whereas the link reaction forms a product which is acetyl coenzyme A. This acetyl coenzyme A becomes the reactant of Krebs cycle. So the Krebs cycle also takes place in the matrix of the mitochondria using the product formed in the link reaction. The matrix contains enzymes needed for the cycle, and we know that there are only two organelles that contain their own DNA. One is mitochondria, and the other one is chloroplast. These are the only two organelles that contain their own DNA. They are independent of the cell division. They can divide, they can form their proteins on their own. These two organelles contain the circular DNA. So where is the circular DNA present in mitochondria? It is present in the matrix of the mitochondria. So we have enzymes for these cycles, that is the Krebs cycle and the link reaction. And then we have 70S ribosomes that are required for protein synthesis and translation of RNA, and several identical copies of mitochondrial DNA. We have circular DNA. What happens that this DNA forms the RNA molecule. This RNA needs to be transcribed. It needs to be transcripted, and for that, mitochondria needs ribosomes, which are 70S ribosomes. These ribosomes transcribe this RNA molecule, and it forms proteins which are required for the mitochondria to work. Then ATP is formed in the matrix by the activity of ATP synthase on the cristae. These, the energy for ATP production comes from the proton gradient between the intermembrane spaces and matrix as well. This ATP can be used for all energy requiring reactions, both inside the cell and as well as outside the cell. So ATP is formed by the activity of ATP synthase. ATP synthase is present on the inner membrane. What happens is, this ATP synthase, it transfers 3 protons inside the matrix of the mitochondria, and then simultaneously it converts ADP plus inorganic phosphate, which are the constituents of ATP. It combines them and forms ATP and a molecule of ATP. So ATP synthase, which is present on the inner membrane, it performs this function that it transports protons from the space between the inner membrane and outer membrane. 3 protons transported at once, and then simultaneously the constituents of ATP, that is ADP and inorganic phosphate, it combines them and forms a molecule of ATP.